Tutorial on movement across a cell membrane. Today we're going to look at what substances can move across cell membranes, how they do it, and the various methods that substances go from one side to the other. Okay, so the role of the cell membrane is to maintain a cell's stable internal environment. It does this by allowing some substances to stay in and others to stay out, okay? And this controlled passage of substances gives the cell membrane its unique properties, such as its ability to be selectively permeable to certain substances. So when we talk about movement across a cell membrane, it is all dependent on the composition of the membrane, being phospholipids, and the surface area available for the exchange of materials. The composition of the membrane being lipid in nature means that lipid molecules can pass through. Things like cholesterol and steroid-based hormones can pass straight through the membrane, okay? Whereas hydrophilic compounds, such as water-soluble molecules, like protein hormones, are unable to pass through the membrane. Other things that are unable to pass through are ions, so charged molecules, and polar molecules, such as water, right, or glucose. These substances require specific channels to pass through, okay? Protein channels embedded into the surface of the plasma membrane, allowing these substances to pass directly through. Okay, so the plasma membrane, due to its selective permeability, okay, means that proteins and ions and water molecules only pass through if the conditions are correct or if the channels are open that allows them to pass through. This differential permeability gives the plasma membrane its key function of keeping the stable internal environment different from the extracellular environment. So when we talk about substances moving across cell membranes, we talk about the following processes: simple diffusion, osmosis, facilitated diffusion, active transport, and when we talk about the movement of bulk substances, it's via endocytosis and exocytosis. So, simple diffusion. It's basically defined as the movement of particles from a region of high concentration to a region of low concentration. Alright? And these different concentration regions, right, the greater the gradient, the more that moves across and the faster this occurs. Okay, so diffusion takes place at a very fast rate if the concentration gradient is very high. If it's low, it'll occur at a slow rate. Alright. Factors that affects simple diffusion are the concentration. Okay, so the concentration of the molecule or the compound that is present on one side compared to the other, the heat that is applied to this region, okay, how small the molecules are. So if the molecules are very small, they'll diffuse much easier than large molecules, and the movement through a gaseous medium. So if it's gaseous, it'll occur much more quickly than it will occur in a liquid medium. Alright. Now, molecules such as O2 and CO2 are regarded as non-polar small molecules, and these diffuse very quickly and easily across the permeable membrane. Okay. Water is a polar molecule, however, it gets away with its diffusion, okay, because it is such a small molecule, alright, but its diffusion rate is far slower than say O2 or CO2. So what we also have is these things called aquaporins present in the cell membrane, and they allow water to pass much quickly and easily into the cell. Alright, ethanol and glycerol are much larger than water, but because they are lipophilic in nature, okay, they can pass through the membrane much easier. Alright, osmosis. It's a special type of diffusion. Okay, it requires no input of energy, alright, and it's defined as water moving from a region of low solute concentration to a region of high solute concentration. Okay, so the movement of free water molecules. That's what osmosis is all about. Now, it does this in this diagram. You can see, right, that water is moving both ways, okay? So the net movement of water is occurring in both directions. However, it is occurring more to the side with more solute, okay? So when we talk about this, it's moving both ways. However, it's moving to the side with more solute, okay, and less free water molecules. So when we talk about osmosis and its effect on plant and animal cells, we talk about three solutions: isotonic solutions, hypertonic solutions, and hypotonic solutions. So, in an isotonic solution, the concentration of water or the concentration of solute is the same on both sides, all right, intracellular and extracellular. Hypertonic solution, there is a higher solute concentration in the side of the cell, therefore the water moves into the cell. Okay, With hypotonic solutions, the solute concentration inside the cell is lower than outside the cell, and the water therefore moves out. Okay, and this is visualized in this diagram here. So this here, right, turgid, right, the H2O is coming into the cell, right, so it's higher solute inside, all right, than outside. Here, flaccid, the cell is remaining stable, H2O is moving in and out at the same rate. So the water molecules are passing in and out of the cell at the same rate. This shriveled cell, okay, H2O is moving out, therefore the solute concentration outside the cell, extracellular to the cell, is greater than inside the cell. So, facilitated diffusion, okay? This is one of the ways that water, large volumes of water, and charged ions can pass in and out of the membrane, and they do so through protein channels. Now, these protein channels are located inside the membrane, and if it's done via facilitated diffusion, there is no ATP needed for this process to occur, okay? So it's not energy. It doesn't need any energy, okay? Now, the transport molecules span the membrane from one side to the other, allowing the molecule to pass directly through. Okay, and there are two types of transport proteins, channel proteins and carrier proteins. Channel proteins, all right, open up and allow the molecule to pass through. Okay, the lining of the channel is hydrophilic, so the water-soluble substances such as ions can pass through it relatively easily. Okay, channels are selective. All right, they only allow certain molecules to pass through. If it's an aquaporin, it'll allow water to pass through. If it's a sodium channel, it'll allow sodium to pass through. It's specific to its specific molecule. Carrier proteins, on the other hand, right, have a conformational change, okay, and they require energy to do it. All right, so to shield the polar or charged groups from non-polar interior of the membrane. So they allow certain substances to move in and out. Active transport. Now, this process requires energy, okay? You need energy in the form of ATP to move substances from an area of low concentration to high concentration, okay? So say, for instance, a cell wants to maintain its glucose, all right? Glucose is a very important molecule. It's used by the mitochondria to produce ATP, and so the cells want to keep their glucose. So the cell will actively transport the glucose back into the cell, even if there is a high concentration of glucose inside the cell, all right? So the active pumping against the concentration gradient. So endocytosis and exocytosis. So in addition to the stuff we've just talked about, if we want to move large amounts of a macromolecule, we do it via endocytosis or exocytosis, okay? So endocytosis is made up of, well, there's three different processes. Penocytosis is the uptake of fluids. Phagocytosis is the uptake of large debris done by macrophages. And receptor-mediated endocytosis requires a specific binding of a protein or ligand to the surface, right? A surface receptor on the cell, allowing it to uptake. And it does so with endocytosis. It engulfs it in the plasma membrane and creates a vacuole, or creates a vesicle, and this vesicle goes into the cell, okay? And it allows the uptake of large amounts of material. So, endocytosis is depicted in this picture here, where the membrane buds inwards on itself. Okay, it pinches off, enclosing the material in a phospholipid vesicle, and this vesicle moves into the cell where it is needed. Exocytosis is just the opposite of endocytosis. Okay, so we have a vesicle produced by the Golgi apparatus. All right, inside that is the bulk movement of, say, a protein or a lipid. This then fuses with the cell membrane, expelling its contents into the extracellular space. So, there we have it. That is the movement of molecules across membranes. Hope you enjoyed. Thanks. Biology Unit 3 and 4 lectures are on sale now. Visit firstclasseducation.com.au.